Welcome back to Cancer is a Mitochondrial Metabolic Disease Basics. Most ketogenic diets focus on one question: how low are the carbohydrates? A more advanced question might be, how low is your GKI, the glucose ketone index? But in cancer metabolism, there may be another question that matters. What type of fat are you eating? Because not all fats behave the same way inside of a cancer cell. Some fats are relatively resistant to oxidation. Others are more vulnerable to lipid peroxidation, the membrane damage that can help trigger an iron-dependent form of cell death called ferroptosis. And that raises a fascinating possibility. Could two ketogenic diets produce similar glucose, ketone, and GKI readings, yet have very different effects on tumor biology because their fat composition is different? Today, we're going to compare saturated, monounsaturated, and polyunsaturated fats, examine how each may influence ferroptosis, and ask whether the right fat profile could make a cancer ketogenic diet more strategically powerful. Let's dive in. So this is the central question of this video that we're going to try to answer. Can fat type change tumor biology on a ketogenic diet? And as mentioned in the intro, there is a tool designed by Seyfried et al. Called the glucose ketone index. And I'm not minimizing the importance of the GKI. It is critical to know how deep into ketosis you are to know whether or not you're maximizing your chances from a glucose, ketone, insulin, etc. perspective. But there's likely more to the story. So I had this graphic created to kind of summarize the importance of a ketogenic diet before we get into fat composition. So by being on a ketogenic diet, you're going to have lower glucose availability to get through these glucose transporters. You're going to have a lower insulin and lower IGF-1 situation, and you're going to have higher ketones. And given that insulin and IGF-1 will signal through PI3K, AKT, and mTOR, some of the most important pro-growth pathways that will set up metabolic reprogramming and lead to resistance of tumors. This is a very important piece of it. Now, when you're able to lower the amount of available glucose to the cancer cells, you're going to have one of two major things happen. You're going to have less cytosolic substrate level phosphorylation, or the making of ATP through the fermentation of glucose. You're also going to have lower flux through the pentose phosphate pathway, or the PPP, which is critical for biomass and NADPH creation, which is important for the cancer's ability to maintain redox homeostasis. And then one of the, I think, more cool and less well-known benefits to a ketogenic diet is that the ketones themselves will actually, in a roundabout way, lower the cancer's ability to ferment glutamine because of the need for ketone breakdown to utilize succinyl-CoA. And that is probably better illustrated in this Seyfried graphic that I added a little bit of context to. So what this is the fermentation of glutamine to ATP. And right here, succinyl-CoA is the step that is going to next generate ATP and have succinate as the byproduct of that reaction. And what I'm trying to say is that when ketones are broken down by normal cells or by cancer cells, it actually steals away part of the succinyl-CoA pool to then divert towards ketone breakdown. And that could take away needed substrate for this mitochondrial substrate level phosphorylation, or the making of ATP without oxygen or without an electron transport chain from glutamine. So pretty amazing things that a ketogenic diet can do. It's not the entire protocol, it's not the entire framework, but it is a foundation to the press pulse ketogenic metabolic therapy proposal. However, what we're going to be talking about in this specific video is the type of fat and how susceptible each type is to ferroptosis. And what we're going to be comparing, and the literature will be comparing for us, is the difference between a saturated fat, a monounsaturated fat, and a polyunsaturated fat. And what it boils down to is that the more double bonds in these fatty acids, the more points of saturation, the more susceptible they are to oxidative stress, lipid peroxidation, and the setting up of ferroptosis. And the difference is actually quite striking when actually studied. So I'm going to use this paper right here that was published in March of 2019. And it was titled Exogenous Monounsaturated Fatty Acids Promote a Ferroptosis Resistant Cell State. And it says here in the abstract, the initiation and execution of cell death can be regulated by various lipids. How the levels of exogenous environmental lipids impact cell death sensitivity is not well understood. We find that exogenous monounsaturated fatty acids, or MUFAs, potentially inhibit the non-apoptotic, iron-dependent oxidative cell death process of ferroptosis. This protective effect is associated with the suppression of lipid reactive oxygen species accumulation in the plasma membrane and decreased levels of phospholipid-containing oxidizable polyunsaturated fatty acids. Treatment with exogenous MUFAs reduces the sensitivity of plasma membrane lipids to oxidation over several hours. This effect requires MUFA activation by acyl coenzyme A synthetase long chain family member 3, ACSL3, and is independent of lipid droplet formation. Exogenous MUFAs also protect cells from apoptotic lipotoxicity caused by the accumulation of saturated fatty acids, but in ACSL3 independent manner. Our work demonstrates that ACSL3 dependent MUFA activation promotes a ferroptosis resistant state. And basically what they're saying is that if there's high amounts of PUFAs, then it's ferroptosis sensitive and the plasma membrane has lipid ROS. And if there's more MUFAs, comparatively speaking, then it's more ferroptosis resistant and there is no plasma membrane lipid ROS. That led me to find this paper that was published in August of 2021 and is titled The Peroxidation of Omega-3 and Omega-6 Polyunsaturated Fatty Acids in the Acidic Tumor Microenvironment Leads to ferroptosis-mediated anti-cancer effects. And what it says in the abstract is that tumor acidosis promotes disease progression through a stimulation of fatty acid, FA, metabolism in cancer cells. Instead of blocking the use of fatty acids by acidic cancer cells, we examine whether excess uptake of specific fatty acids could lead to anti-tumor effects. We found that omega-3, but also remarkably, omega-6 polyunsaturated fatty acids, PUFA, selectively induced ferroptosis in cancer cells under ambient acidosis. Upon exceeding buffering capacity of triglyceride storage into lipid droplets, omega-3 and 6 peroxidation led to cytotoxic effects in proportion to the number of double bonds, and even more so in the presence of diacylglycerol acetyltransferase inhibitors, DGA-TI. Finally, an omega-3 long-chain PUFA-rich diet significantly delayed mouse tumor growth when compared with a monounsaturated fatty acid-rich diet, an effect further accentuated by administration of DGA-TI, or ferroptosis inducers. These data point out dietary PUFA as a selective adjuvant anti-tumor modality that may efficiently complement pharmacologic approaches or even dietary ketogenic approaches. So this graphic was pretty interesting, and basically what they're saying is that tumor fatty acid type matters, but also the pH matters of each compartment, and we're going to dig into this. So let's head to one of the most interesting experiments that these researchers carried out in this paper. And that was basically looking at what happens after 24 hours of exposure of certain fatty acids on oxidative stress accumulation at both physiologic pH and acidic pH. And just for context, you may be asking yourself, why does it matter and why do we care about the pH? The reason why we care is because our body is tightly regulated to a pH of about 7.35 to 7.45. That's called physiologic pH. And our body tries everything that they can do to maintain that pH so everything works like it's supposed to. But inside the tumor, in the tumor microenvironment, it is extremely acidic compared to that physiologic pH. So the pH is much lower. As a matter of fact, it ranges from 6.0 all the way to 6.5. And that changes the ability for the immune system to work, whether therapies are able to get to the tumor itself, and it leads to a pseudohypoxic state that leads to metastatic spread, et cetera. So that's why they did a study at both 7.4 and at 6.5. So I have created a table that breaks down some of the major types of fatty acids on this table, which they conducted experiments on. And I have listed not only the name of what each acronym actually means, but also the type of fat it is and the relative amount of oxidative stress markers for each type of fat at both pHs tested. So let's start with palmitic acid, which is a saturated fatty acid. It has, relative to the rest of the fatty acids, 1x at both physiologic pH and the acidic pH found around tumors. If we compare that to a monounsaturated fatty acid, it actually has about the same, slightly more resistant to oxidative stress, and that would be at 0.9x respectively for both pHs. It gets interesting because when I was reading this, I was expecting omega-6s, such as arachidonic acid, to be basically the same as omega-3s. And what I found was actually fairly shocking because at physiologic pH, they actually are fairly similar. Here is arachidonic acid, omega-6, and then EPA and DHA, the two major long-chain omega-3s, 2.7 and 3.3x respectively, compared to the saturated fatty acid, palmitic acid. But where it gets very interesting is actually when the pH drops to 6.5, it becomes more exaggerated. So we see here that at a tumor type pH of 6.5, the omega-6 is about 3.5 times more likely to have oxidative damage associated with it. EPA slightly more at 4x, but then what's fascinating is DHA, one of the main long-chain omega-3s, is 7 times more likely to participate in lipid peroxidation than its other omega-3, omega-6 counterparts and the monounsaturated and saturated counterparts. Pretty astounding. Now, I pulled this study because it showed what happened to these mice and rats when they actually gave different types of fats to these animals. I'm going to focus on the two bottom pieces of this table, H and I. And we see here that when they were given a diet that was heavy on the oleic acid or the monounsaturated fatty acid, because of the increasing ferroptosis resistance, or that's what they believe, you see that the tumor exponentially goes up. But when the mice are given DHA, the omega-3 fatty acid that is the most ferroptosis sensitive, you see a much different story. You see at the end of, let's just make it apples to apples, at the end of 35 days, the tumor volume goes from about 800 down to less than 400. So about 50% reduction in tumor size at the end of 35 days. It would have been nice to see what actually happened to these mice if they went down further, but I'm not sure if they died or what happened after 35 days for the oleic acid. And then they gave an additional inhibitor for this red, and you saw how there's like a synergistic effect between the type of fatty acids and a metabolic inhibitor, this DGAT1 inhibitor. Now, then in I, they're looking at what is the percent survival of each of these mice, depending on the type of fat they got or the inhibitors they got. And I guess I kind of answered my own question here because I was wondering what happened to these mice after 35 days. And it seems that a lot of these mice are dying within 6 days post-treatment if they got a heavy oleic acid diet. Whereas if they got DHA, they lived not quite twice as long. And if they got DHA plus this DGAT1 inhibitor, they lived about 3 times as long. So pretty unbelievable difference. Again, same mice, same tumor. They either got PUFAs in the form of DHA or long-chain omega-3 fatty acid, or they got MUFAs, which is a more resistant fat to ferroptosis. Now, this was actually one of the more interesting experiments they did. So they either gave these mice, and we're going to look at their tumor volume and the survival endpoints for these mice with 2 separate additions to the DHA. So they gave the most ferroptosis sensitive fatty acid, the long chain omega-3 DHA. And we see that the tumor volume is, let's just estimate it maybe 500 at the end of 35 days. If they gave a ferroptosis inhibitor, this ferrostatin 1, you saw that the tumor volume went significantly up. And if they added a glutathione blocker, in this case sulfasalazine, by blocking SLC7A11 or XC, the cysteine glutamate antiporter, you see that there was a synergistic effect between the DHA and blocking glutathione. You saw a tumor volume that was lower than what happens if you just gave DHA alone. And then we see here that post DHA, they lived about 20 days before almost all of them had passed away. And then if they got sulfasalazine plus DHA, they lived for significantly long. I wish they would have put this out further to see how long they actually did live compared to the controls. But it seems to be a significant difference when given DHA and sulfasalazine, not only with the tumor volume, but also the actual survival of these animals. And then when we looked at if they got DHA, the percent survival was longer. I'm guessing it would have rapidly gone down looking at how this one went because it only maxed out at 16 days here. But we see that if they gave a ferroptosis inhibitor, this ferrostatin 1, they rapidly died compared to those who did not get the ferroptosis inhibitor. And this is kind of a roundabout way of looking at, basically things that inhibit ferroptosis. We've talked about these things in the past, and some of these things have taken some heat on, like the astaxanthin or NAC or glutathione. But this is basically what it's showing. So here they're taking away glutathione and adding DHA. And in this case, they're actually not necessarily directly stimulating glutathione, but probably in some ways they're kind of boosting the antioxidant response, and we're seeing what happens to the tumor, right? There's a growth effect or an inhibitory effect, depending on the kind of antioxidant status, because these things are either allowing buffering to happen or buffering to be impaired. So that way that DHA can be peroxidized and allow for ferroptosis to happen. And again, this is just, I wanted to highlight this. Sulfasalazine has been in all of our classic glutamine and ferroptosis kind of graphics that we always look at this graphic, and sulfasalazine right here. It actually directly inhibits this SLC7A11, also known as XC, and also known as, in some publications, the Achilles heel of the tumor. So pretty cool that they actually looked at not only DHA versus other fatty acids, but they actually added the metabolic inhibitor sulfasalazine to this experiment to see what actually happens. Now, I pulled these papers just because, why not? So interestingly enough, polyunsaturated fatty acids may also be more ketogenic. These are a couple of papers showing that if you were to compare monounsaturated versus saturated versus polyunsaturated, typically the polyunsaturated fatty acids actually tend to be more ketogenic. So it kind of goes back to the GKI, right? If they enhance ferroptosis more and change the susceptibility of these cancer cells, and they have an anti-inflammatory effect that we all know that omega-3s have, and it can maybe even enhance your GKI, it seems like this would be a net win. Now, the amount of omega-3s that you may want to take is going to be something probably going to be discussed between you and your doctor. I'm not going to give you prescriptions here, but there is something called an omega-3, omega-6 ratio, and there's something called an omega index that's easily checked at pretty much any lab, including Quest, and I use it regularly. If you like videos like this, please like, share, and subscribe. And until next time.